In this video, we're going to talk about the difference between protostomes and deuterostomes. If you remember from the embryonic development video, after the blastula formed, gastrulation happens. And you can see that on this picture here, where part of the blastula is starting to pinch in. And this pinching in is forming a structure that will ultimately turn into the gut or the digestive tract. So this pinching in area is called the blastopore, and eventually it's going to go all the way through. So we're going to end up with a kind of a structure that has this hole going through it. So that's a bad drawing. So it's kind of like a donut, if you will. So it's still all connected, but there is a pathway or a passageway going through the middle of it. So this blastopore is the pinching in part, and it eventually extends all the way to the other end of the structure. So most animals have openings at both ends of the digestive system or the digestive tract. So at one end you have the mouth where the food comes in, and at the other end you have the anus, which is where waste products come out. Now in bilaterally symmetrical triploblastic animals, they can be either protostomes or deuterostomes. And the main difference here is whether the mouth forms at the blastopore or whether the anus forms at the blastopore. So if we take a look at the formation of the mouth, so in protostomes, the mouth forms first. So the blastopore, that pinching in here, is ultimately going to differentiate and become the mouth. So on this other picture here, the blastopore would be this side here. So this would be where that initial gastrulation happened, and then ultimately the blastopore went all the way through the blastula in order to create this tract or this hole going all the way from one end of the organism to the other. Now in deuterostomes, the mouth is the second part that is produced. And so the blastopore, that pinching in area, forms the anus first, and then it continues to kind of pinch in until it forms the mouth on the other end. So protostomes, the blastopore is going to form the mouth first. In deuterostomes, the blastopore is going to produce the anus first, and the mouth is going to be the other end when that gastrulation connects to the other side of the blastula. So some examples of protostomes are going to be arthropods, which are insects, mollusks like snails and scallops, and annelids like earthworms. For deuterostomes, we have echinoderms, which are things like sea stars and urchins, and chordates, which includes things like mammals, including humans, and reptiles and amphibians. Protostomes and deuterostomes also differ in how the mesoderm forms. And remember, the mesoderm is also going to ultimately form the coelom or the body cavity. So in protostomes, we have schizocele, which means that the cells in the blastula are going to form the mesoderm in these two clumps. You can kind of see that here. And then the coelom, or that body cavity, forms inside the mesoderm. In deuterostomes, we have enterocele, which is where the endoderm pinches off, which we can see here, to form the mesoderm. Protostomes also have something called spiral cleavage. And remember that cleavage is just that embryonic cell division or mitosis. So it's that rapid mitosis that happens after the zygote is formed as the embryo is developing. And spiral cleavage means that as these cells are being made, they're not lining up right on top of each other. So they're actually kind of shifted a little bit, forming this kind of, I guess, more of like a spiral or kind of an offset kind of cell stacking, if you will. With deuterostomes, they have a radial cleavage, and so the cells are going to be more stacked on top of each other, like this. And another difference between protostomes and deuterostomes. In protostomes, they have what is called determinate cleavage. And this has to do with the differentiation of the embryonic stem cells. So remember that we talked about how these embryonic cells So we talked about how these embryonic stem cells are undifferentiated, but in protostomes, pretty much as the cells are going through mitosis, their fate is being determined very early on. And so they don't really have these true embryonic stem cells because they are already starting down the pathway of what kind of cell they are ultimately going to become as they are being formed. With deuterostomes, they have the indeterminate cleavage, and that's where we have these totipotent embryonic stem cells. And so these are stem cells that they're not kind of predetermined as to what cell they're ultimately going to differentiate into yet. So that differentiation occurs later on in the embryonic development.